Multiple choice. 2018 paper, the second half of the multiple choice. Okay, question 11. A field trial was carried out to investigate the effects of massive phosphate fertiliser applied on the growth of barley. The barley was planted in plots of equal area on a hillside and fertiliser applied as shown in the diagram. Which of the following procedures would improve the field trial design to take into account higher soil moisture levels at the bottom of the hill? Okay, can we get rid of D straight away? Because that's kind of obviously wrong, hopefully. You would see that just picking another hillside is not going to get around the fact that we have higher soil moistures at the bottom of hills, because it would surely be the same problem on another hill. So D is not in there. Okay. C is also not in there. I'm just working my way up the way now. Increase the number of plots. Well, if we kept going on this hill, so this would be another 2, that would be another 4, that would be another 6. That's just building in the same problem again and again. So it's not increasing the number of plots. Okay. It's also not A, increasing the range of phosphate fertilizers. I mean, if I said, right, okay, so instead of having this the 2, I'm going to split this one and I'm going to have a 1 and a 2 and 3, 4. Again, I'm not dealing with the problem of there being more moisture at this end than there is at this end. What will do that is if I randomise the treatment plots. So that means that maybe on this hill, you have a 2 here, a 2 here, and a 2 at the bottom. Okay, and then a 4 in here, a 4 in here, and a 4 at the bottom, and then a 6, and the 6, and the 6. And that means that what we've got here is taking away from your hillside issue. Okay. Question 2, which of the following is an example of kin selection? Okay, so what I'm looking for here is individuals which are helping the survival chances of related individuals so that you have more chance of shared genes being passed on. Okay, right, so worker leafcutter ants raising young ants in their colony. In their colony? No idea what that word is. Colony. Okay, that is correct straight off. Okay, so worker ants, remember, they are not actually providing offspring themselves, but if they are helping to raise younger ants in the colony, all of those ants will have come from the same gene pool, very narrow because it will be one queen. So effectively you're talking at siblings here, and so that means that they're helping for their shared ancestry to kind of keep going. Okay, vampire bat regurgitating blood to feed an unrelated bat. That's an example of altruistic behaviour. We've got a dominant lion feeding on a zebra kill before its offspring. That's not really helping its offspring for a start. That's not helping things to be passed on. But this would be dominance hierarchy. And a young orangutan spending a long period in parental care to learn complex social behaviours is exactly what it says. They're spending a long period in parental care so that they get all the complexity of social behaviours. But it's not about a related individual increasing survival rates. Okay, question 13. An experiment was carried out to investigate the growth rate of pigs. They were put into 5 groups of 8 pigs, each with the same average initial body mass. Each group was fed a diet which contained either 0, 10, 20, 30 or 40% of faba beans. The pigs were reweighed each day for 40 days. Which aspect of the experimental design increased reliability of the results? Reliability. I am looking for how they are repeating. Okay, I'm looking for internal repeats or full set repeats. Okay, 5 groups of pigs were used. Well, that's not reliability of the results, that's how much of a spread of information they had, because that was your 0, 10, 20, 30 and 40. So that's to do with the validity of design, it's not to do with reliability. Okay. Pigs were reweighed each day for 40 days. Again, that's experimental design, but that doesn't change about the reliability of it. Each group had the same average initial body mass. Again, that's about your setup. Each group containing 8 pigs, however, I have now got an internal repeat running. Okay, question 14. We've got an investigation carried out into social hierarchy in a group of 5 hens. Hens established dominance by pecking each other aggressively. The number of pecks given and received was recorded. Results are shown in the table. Okay, so someone spent some time just watching chickens. Okay, right, number of pecks given, and then we've got the number received down here. Okay, the green's maybe not the best colour. Let me just swap it back to yellow. Okay, right. So if I'm looking for the most dominant hen, what I'm looking for is something that basically did not receive any pecks, because everyone else was just like, no, not going for them. And there you go, look at Y. And not only that, the number of pecks given, Y was quite happy to just attack everybody else. So Y is decidedly the most dominant. Okay, right, and then if we look for the other end of the spectrum, okay, I feel quite sorry for Z. Here's Z. Z did not peck anybody else, but however took a battering off everyone. Okay, so the low kind of level of this one is Z at the far end. Okay, so we've got most and we've got least. So to fill in the blanks in between, what we're looking for is kind of the next levels of that. So if I'm looking for next level for least dominant, okay, you can see quite clearly here that X only pecked Z. So Z was at the very, very bottom, and X was not much better, okay, because X still got attacked by all the rest of them, okay. And then we're looking for the next one up again. So we're going to W for the same logic and V for the last. Okay, so Y, V, W, X and Z. Y, V, W, X and Z. Each type of human cell has a different structure and function because, okay, pretty much a straight KU to be honest. It's because the only some of their genes are expressed. It can't be because they contain different genes, because the whole point is that your DNA is everything. It's the same in every cell. We don't lose genes. We just keep them turned off. And we don't gain genes. This would all be quite scary actually, the C and D stuff. Okay, it is just that we pick and choose which genes are expressed. Okay, question 16. The list describes some uses of stem cells. Studying how cells differentiate, researching the development of Parkinson's disease, producing skin for skin grafts, bone marrow transplants. Which of these uses are not therapeutic? So therapeutic means that we're looking at something that is being used medically. Okay, so this is not medical, studying how cells differentiate, and this is not, although it might become something that is used medically, but we're researching development of a disease is not a therapeutic use, because it's not being used to treat somebody. Okay, so 1 and 2, so B. Okay, question 18, a speciation one. Population of finches became isolated on an island. The graph shows the range of beak sizes within the initial population at the time of isolation and in the population after many generations. Okay, so our initial population is our solid line, and after many generations we've gone to dotted. Which row in the table shows the type of selection pressure and the type of speciation? Okay, so you have two decisions to make. One is this directional selection or is this stabilizing? And is it allopatric or sympatric speciation? Okay, so directional selection means that if this is your starting mean, you have to shift the whole curve. Okay, either this way or this way, but you need to move the whole curve. It is not directional. What you have here is stabilizing selection. So you've started off with your range, and then you've selected for this bit in the middle, or you could argue you've selected against these kind of top and bottom. So what you've ended up with is this bit becomes, you can see here, kind of a narrower range in the middle. So it's stabilizing. Okay, then you need to decide whether it's sympatric or allopatric. You need to go up to here. Population of finches became isolated on an island. That is geographical isolation, and that is allopatric. So C. Question 20. The analysis of DNA sequences from different organisms is used in the production of molecular clocks. This analysis is based on the assumption that over time DNA sequences undergo mutations at a constant rate. Okay, it is still assumed that they will be random, that they are spontaneous, but the whole point is that it's the constant rate. Okay, and if it was a varying rate, it could hardly be a clock. The tick would not work.